I have the great pleasure of talking to you about my absolutely favorite topic, which is diatoms that live on aquatic animals. I've been working on this topic for about 15 years now, and I have long been thinking about the best way to present this topic to you today. Eventually, I decided that I would simply tell you a story. I will mention some studies here and there, but overall, I will focus on theories and patterns that at this point in time are supported by only fragmentary evidence when it comes to published data, but also some of my own observations and experiments that I have not yet published. It's the Diatom Web Academy, so I assume the people watching us do know diatoms pretty well. In this presentation, I will talk a little bit about diatom taxonomy, but the main focus of the talk will be on animal-associated diatom ecology. So let me very quickly list a few well-known facts about diatoms. Like the vast majority of algae, diatoms are mainly autotrophic, although heterotrophic or mixotrophic taxa also exist. At the moment, there are about 25,000 diatom species known, but we expect there are at least 10 times more that are still waiting to be discovered. And of course, species delimitation in any microbial group is not that straightforward anyway. So maybe let's just say that it is extremely likely that there is a high number of diatom species and forms that have not yet been discovered, especially when it comes to the marine ecosystem. Diatoms live on every continent and in every ocean. In fact, they live everywhere as long as minimal levels of moisture and sunlight are provided. There are freshwater species, brackish and marine species, and species that live in soil, on the tree trunks, on mosses, in rock fissures, and crevasses of any kind. Also, there are species that live in cold and hot deserts. There are planktonic diatoms that live suspended in a water column, but there are also very many different types of surface-associated diatoms. Not necessarily benthic, as benthic implies organisms associated with the bottom, which is rather limiting. So here I mean diatoms that require hard surfaces and attachment points to grow. For example, there are diatoms that live on seaweeds and aquatic plants. There are diatoms that live on other diatoms. And there are diatoms that live on aquatic animals. And these diatoms are called epizoic. There are epizoic diatoms on barnacles, snails, sponges, bryozoans, and copepods. And actually, very likely, any aquatic creature living within the photic zone and possessing appropriate stable body surfaces will be covered in diatoms. So I will first show you some examples of diatoms growing on animals, and the list will not be exhaustive, unfortunately. And I will later discuss why some animals do host diatoms and some do not. I will explain more about what those stable, appropriate body surfaces are. And I will also say a few words about the host specificity in epizoic diatoms. So let's look at some interesting examples. And let's start with invertebrates as hosts for diatoms. Daniel Wujek observed diatoms growing on mayflies. Immature mayflies, the so-called naiads, are aquatic. They live underwater, especially running water, for 6 weeks to 3 years. During that time, they will go through about 21 molts as a naiad before they become winged adults. So Wujek observed that 3 diatom species dominated the naiads, even though many more diatom species were present in the periphyton of the same creek. In other words, while the diatoms dominating mayflies included species that were abundant in the water body where the mayflies lived, mayflies were inaccessible or were not the preferred substratum for many other periphytic diatoms. Quite interestingly, diatoms were found on the naiads from the family Canidae, but not on those from the family Ephemeralidae. We do not know why it was so, and the author does not make any suggestions. One of the possibilities could be that the two families have different molting cycles, different behavior, or perhaps it is about the different microtopography of their cerci or cerci, depending on what your Latin teacher prefers. Diatoms are relatively often reported from copepods, which are very common small crustaceans in the zooplankton of both fresh and marine waters, where they are important members of aquatic food webs. Even though diatoms are often observed on copepods, most copepods, if you go out sampling for copepods specifically, will not host diatoms. Why so? That may again be related to the molting cycle of different species. In some groups or species of copepods, molting will occur at short or very short intervals, perhaps too short for diatoms to develop viable colonies. On the other hand, as I have already mentioned, copepods are a very important food source for very many aquatic organisms, and they often do not live very long, simply because they are eaten by almost anything. Several authors have observed that whenever there are diatoms on copepods, the community is composed of a single species. It's not clear why it is so, but the principle here may be similar, or in fact the same, as in the founder effect. The entire community on a given individual is established by a single diatom cell that reproduces very fast and whose daughter cells claim all available attachment points. Diatoms reported from marine copepods so far belong to mainly a few genera. Those genera include Pseudohumanthidium, Falcula, Protoraphis, Steptornema, Ligmophora, or Tabularia. All of those forms are erect forms. And the situation seems to be similar when it comes to freshwater copepods, which is the erect, araphid, tychoplanktonic diatoms often dominate the surfaces of copepods. Are these forms specific to the host species or the host group, the copepods? Well, in freshwater habitats, the answer seems to be no. But in the marine ecosystem, the answer is we are not sure. It is highly possible that diatoms associated with marine zooplankton do have adaptations that allow them to quickly colonize the small floating crustaceans. However, it is not clear whether these diatoms occur only on copepods. We still need some more research to confirm or refute those hypotheses. There are diatoms on other crustaceans as well, from mobile crabs, lobsters, and shrimps to sessile, barnacles, and crustacean fish parasites, for example. So let's look at some challenges the Epizoic diatoms must overcome to develop viable colonies on crustaceans. For example, arthropods periodically shed the exoskeleton during molting, which physically removes any attached biofilm. Frequent molting, especially in juveniles, creates an ephemeral substrate that prevents long-term folding. In early life stages, when the molts occur rapidly, only a few diatoms, if any, can accumulate before the exoskeleton is shed. So, of course, this reduces the Epizoic diatom diversity very efficiently. Only after crustaceans reach a terminal mold, which is adulthood, when molting stops or slows down, the carapaces become stable enough for dense or denser folding communities. For example, adult blue crabs and lobsters often carry rich epibiotic assemblages, including diatoms, while fast-molting juveniles remain clean. So this cycle selects for diatom species that can recolonize quickly between molds or persist in protected microhabitats, like, for example, crevices around articulations. If a crustacean exhibits sex or age-specific molting patterns, it can lead to corresponding differences in epizoic diatom colonization. What is more, many crustaceans engage in grooming behaviors. They often have specialized appendages to scrape off the biofilm. And naturally, again, that will also affect epizoic diatoms. Many aquatic invertebrates undergo metamorphosis or stage changes. These transitions often involve shedding larval cuticles or developing new body surfaces. Such life history traits reset epibiotic communities at each stage. Any diatoms on a larval exoskeleton are lost when the larva molts or when the metamorphosis happens and the host animal transitions into the next stage. That means that diatom colonization must begin anew on the juvenile form. Consequently, diatom communities may differ between life stages, and specialist diatoms must either disperse to new hosts or tolerate the transition. In some cases, larvae present very short-lived or smooth surfaces that are inhospitable to diatoms. In those cases, the epizoic colonization only begins in later life stages when the exoskeleton is fully formed. Sponges, hydroids, and bryozoans. These sessile invertebrates often produce chemical antifolding defenses that can very efficiently reduce microbial and algal growth on their bodies. For instance, marine sponges are known to secrete bioactive metabolites, sometimes through the symbiotic bacteria. Those metabolites suppress diatom settlement on their surfaces. Diatom density is often significantly lower on sponges than on any other substrate located in the same system. Compounds produced by sponges and hydroids can inhibit diatom attachment and therefore alter significantly diatom community composition, often lowering species richness, as not many opportunistic species will be adapted to withstand this antifolding mechanism. And similar is observed from some bryozoans and echinoderms. Echinoderms are sea stars and sea urchins. Those remain largely free of microfouling. So these chemical defenses mean that only certain tolerant diatom taxa or no diatom taxa at all can persist on such hosts. When diatoms do occur on chemically defended invertebrates, their communities may be relatively low in abundance and diversity, or be present only in small patches where defense is locally inactive. Some diatoms may adapt to these conditions, for example, by developing resistance to the host deterrent chemicals or by timing the colonization so that the colonization can happen when host defenses are compromised. Some reports indicate that diatoms, both centric and pennate forms, can live inside the tissues of sponges. The nature of that relationship is not well understood. One would think that shells, such as those of snail shells, would provide a perfect habitat for diatoms, as they very much resemble rocks, and rocks are generally suitable for diatom growth. A few of the existing studies, however, showed that both diatom abundance and diatom species richness are rather low on gastropod shells. As shells contain a high concentration of calcium carbonate, it was hypothesized that the chemistry of the shell alone may deter many diatom species. On the other hand, it was also observed that snail shells are generally exposed to very intense grazing by other snails, often snails belonging to the same species. That intensity may change seasonally or show different patterns in invasive species. Some gastropods also secrete mucus over their shells, which facilitates cleaning and prevents attachment. Of course, there are also invertebrate groups that will, with high probability, not host any diatoms. Groups that come to mind would be the soft-bodied mucus-covered animals, such as jellyfish and anemones that continuously slough off particles and shed sheets of mucus. So invertebrates host with dynamic surfaces due to frequent molting or shedding and generally short life cycles tend to have sparse, transient Epizoic diatom communities. In contrast, those hosts with more stable surfaces, like those with long-lived exoskeletons or shells without a frequent shedding, can develop richer diatom assemblages. The ephemeral nature of frequently molded exoskeletons reduces the number and density of following species on many mobile invertebrates. This dynamic strongly influences diatom community structure, like fast colonizing, fast reproducing diatoms may dominate short molting intervals, while the slow-growing diatoms will not be able to establish viable colonies, being frequently sloughed off. By contrast, on an invertebrate that doesn't regularly renew its surface, for example, a horseshoe crab in terminal mold, the more complex, more diverse diatom community has time to develop. Okay, let's now move to vertebrates. Do fish host diatoms? That's a good question. Most fish are coated with a layer of mucus secreted by epidermal cells. This slime layer is rich in antimicrobial enzymes that are very good at inhibiting microbial growth. For example, extracts of pufferfish skin mucus showed good antifouling activities in various assays, preventing the settlement of both bacteria and microalgae, including diatoms. In fact, the crude fish mucus extracts are currently being explored for their very strong antibacterial and antifouling properties. So we can imagine that for diatoms, that thick mucus will act as both physical and chemical barrier. Diatom cells landing on a fish may become embedded in the mucus, but it does not mean that those diatoms live in the fish mucus. They likely live there only for a moment, but diatom shells may be found in the mucus also after the diatoms have died. That's why we should not call those diatoms epizoic. It is rather a mixture of whatever is present in the river, lake, or the sea habitat. There are some studies that claim to explore epizoic diatoms on fish. For example, Elisa Velasco and colleagues claim they explored epizoic diatoms on a very slimy, as the authors describe, catfish. The authors brushed 66 individuals of a certain invasive catfish species, finding more than 260 diatom species. The authors observed that these diatom species were highly representative of the diatom flora previously found in the rivers from where the fish were collected. So already the fact that we have a very high number of diatom taxa on those 66 individual fish indicates that the collection of diatoms is likely pretty random, and that there are no species that would prefer the fish substrate over other substrates available in that water body. In other words, there are no diatoms that would thrive and not only be present on those fish hosts. So, of course, if a living diatom cell can get trapped in the fish mucus and the fish is mobile, those animals may act as vectors for the various diatom species, yes. But I would be very careful with calling those species epizoic. At the same time, I'm not saying that diatoms cannot live on fish. The effectiveness of mucus in deterring diatoms will be different in different fish species or fish groups. Fish or fishes with copious viscous slime, like eels or hagfish, are free of visible biofilms, but fishes with much lower mucus production may accumulate some epiphytes on their scales. The mucus keeps the fish surface hydrodynamically smooth, so it creates that constantly moving non-solid substrate. So epizoic diatoms on fish would have to tolerate living in the fish mucus or perhaps be able to cling to the scales that stick above the mucus layer in some fish taxa. And because living in the fish mucus is not the same as living in diatom-produced mucus, right? Those two liquids or gels have very different chemical compositions and very different functions. Hypothetically, some epizoic diatoms may be able to use some temporary breaches in the mucus layer to attach before the mucus is replenished. Those breaks in mucus production may occur, for instance, if the fish is stressed or is suddenly placed in much cooler water. Unlike some reptiles, fish typically do not shed the entire skin at once. However, certain species do slough off patches of epidermis periodically as a fouling control. Some benthic fish, like scorpionfish and stonefish, shed their skin 2 to 3 times a year to eliminate epibionts. The skin of those fishes is shed in patches or sheets, and it is unusual behavior for fish, but it is very effective strategy of self-cleaning. These episodic sheds dramatically reduce diatom load, of course, on the fish, and of course, once the process is finished, the epizoic diatom community is reset. The reef stonefish that you can see in the picture here becomes bright pink after shedding. As a result, only diatoms with particular life strategies can recolonize between shedding events. Those fast attaching, fast reproducing diatoms can possibly bloom in the window before the next skin slough. Of course, the shed skin itself can carry the diatom assemblage away in the water column. And what is the importance of that dispersal at the moment, we do not know. So when it comes to diatoms on fish, some species may host diatoms that would actually grow on their skin, but most fish species would not carry such communities. Since the major obstacle here is the continuously excreted mucus that often shows strong antimicrobial properties, it is possible that diatoms would occur in some spots on the fish body that are not affected by the fish mucus. There are many interesting examples of fish with perhaps unusual structures on their body and some innovative anti-folding strategies, but I do not think we have time today to focus so much of our attention on fish alone. Let's now look at aquatic birds. In 1982, Kroll and Holmes observed diatoms on the contour feathers of some diving seabirds, such as common murre, as well as Arctic loon, and I think red-throated loon. Only about 25% of the sampled birds had diatoms on their feathers, and only certain spots of the birds' bodies were colonized. The distribution of diatoms on the bird feathers did not show any strong patterns, but the diatom community was species poor, suggesting that only some species out of those present in the water can colonize bird feathers when favorable conditions arise. The feather-associated taxa included species belonging to genera such as Pteroncola, Synetra, and Nagumea. After the two rather short papers by Kroll and Holmes, which were published in 1982 and 1984, no study would look at bird-associated diatoms in so much detail. So again, let me speculate a little bit, always in the context of the health, ecology, and physiology, and using some fragmentary evidence that is available here and there. Aquatic birds, such as ducks and cormorants, are generally not heavily colonized by diatoms, thanks to both physical and behavioral defenses. Their feathers are coated with oil from the so-called preen gland that makes them highly hydrophobic. So that water and any microorganisms in it rolls off easily. A diatom cell in the water has difficulty adhering to a feather because of this water repellent layer. What is more, birds actively and meticulously preen their feathers, literally combing them with their beaks to remove debris, parasites, and any beginning biofilms. This constant grooming usually dislodges the would-be colonizers before they can form a stable community. However, as I mentioned before, in some documented cases, diving seabirds have been found with diatom films on their feathers. The bird species that did host proper communities of Epizoic diatoms spent extended time submerged in productive waters, which can allow diatoms to attach if the bird's preening does not keep up. The diatoms on the bird's feathers were rather small and stuck to feather barbs. Nonetheless, such fouling appears to be relatively rare and transient, often noted in birds that were sick, heavily burdened by mold, or in cold water where preening oil production may be reduced. Healthy birds replace their feathers periodically through molding, usually on an annual cycle. When a bird molts, all feathers and any diatoms on them drop off, and new clean feathers grow in. This annual reset limits long-term diatom persistence on birds. At the moment, the consensus seems to be that any diatom taxa found on feathers are likely generalized freshwater or marine species that temporarily attached rather than obligate feather dwellers. The occasional presence of diatoms on bird feathers has ecological implications beyond the bird itself. Water birds can serve as vectors for diatom dispersal. Studies have shown that diatom cells adhering to duck or swan feathers remain viable and can inoculate new water bodies when the bird flies somewhere else. So while birds usually evade fouling, when diatoms do stick, the relationship is often one of accidental transport rather than a stable epizoic community. I'm moving to aquatic mammals now because I want to leave the sea turtles for the last but not least. Aquatic mammals, which is cetaceans, whales, and dolphins, pinnipeds, seals, sea lions, and walruses, and sirenians, manatees, and dugongs. Cetaceans have mostly bare skin, no fur except tiny hairs. Their primary defense against fouling is an exceptionally high skin turnover rate. For example, some reports inform that a bottlenose dolphin replaces its outermost skin layer roughly every 2 hours. And this is an astonishing sloughing rate. It's roughly 9 times faster than in humans. This constant flaking and renewal ensures the skin stays smooth, which reduces drag, a very important aspect for the fast swimming dolphins. The fast sloughing rate also prevents most biofouling from gaining a foothold. So most of the diatoms are shed before they can firmly attach or multiply. Additionally, dolphin skin is rich in collagen and elastic fibers and hosts a rich community of host-specific bacteria that may also keep microalgae at bay. The result is that small, fast cetaceans like dolphins and porpoises are usually observed to be free of visible algal films. However, specialized diatoms do live on cetacean skin, even on dolphins, but especially on the larger, slower moving whales. In colder waters, whales can develop a noticeable coating of diatoms. Early whalers noted that blue whales in the North Pacific often had a yellowish-brown film on their bellies, and that is why they called those whales sulfur bottoms. We now know that sulfur bottoms are caused by dense species poor or even monospecific biofilms of whale-associated whale-specific diatoms. More recent studies have found that when whales spend extended time in frigid high-latitude seas, the skin replacement slows down. That's because blood flow to the skin is reduced to conserve heat. During these periods, diatoms accumulate in a thick film on the whale's body. For instance, Antarctic killer whales often show a yellow color due to diatom cover, indicating that they haven't lost skin in a while. This fouling is not permanent. When whales do migrate to warm tropical waters, they take a spa day. The warmer conditions restore skin metabolism, and the whales undergo a major molt, shedding old skin and the diatoms with that skin. Some researchers have proposed that one evolutionary driver for long-distance whale migrations is indeed skin maintenance. Whales feed largely in the polar regions, but travel to warm seas to molt and slough off the Epizoic growth. Once the old skin is shed, the whale returns north or south, depending on the hemisphere, with a clean skin. This cycle profoundly affects diatom persistence. Only diatoms capable of rapidly colonizing whale skin or surviving in dormant forms in warm waters can reestablish each year. Some diatom taxa appear to be cetacean specialists. For example, the genus Venetella, Epiphyllina, Plumosigma, and some of the Turciocula species. Turciocula is in fact named after Tursiops, which is a dolphin, because it was first found on a dolphin. Some Turciocula species are apochlorotic and may derive nutrition from either the host skin or, which is maybe more probable, the manatee-specific bacterial flora. Tom Frankovich and colleagues described several Turciocula species from Florida manatees, and all those species together were the dominant group on the analyzed hosts. The loss of chlorophyll in these diatoms suggests they have adapted to either low light conditions, perhaps living deep in skin creases, or to a nutrient-rich symbiotic existence on the host. It is expected that these diatoms will have special attachment mechanisms to cling to skin that is otherwise constantly renewed. Perhaps those mechanisms involve attachment substances that are not affected by skin enzymes. It could also be that specialized animal-associated diatoms reproduce synchronously with the whale's behavior. For example, blooming when the whale is in cold water, and then forming resistant spores when the whale enters warm water to molt. Here we are again in the speculation zone, as not much is known about the survival strategies of these diatoms. And since I am talking about sea mammals, let me say a few words about pinnipeds as well. Pinnipeds, seals, sea lions, and walruses have a fur coat, pelage from French, often with a layer of protective oil, and they split their time between water and land. The pinniped physiology and behavior, such as hull out behavior, which is getting out of water to rest, as well as molting, is quite effective at limiting long-term diatom colonization. In pinnipeds, the diatoms that do appear on fur are likely those that also grow on nearby rocks or kelp, simply transferring to the animal body during contact. However, it is also possible that some body parts of seals may harbor some more specialized diatoms. Those body parts could be whiskers and claws. And again, I'm kind of making this up. There is currently no evidence to support my hypothesis, but personally, I think it is possible that we can find some interesting diatoms on animal whiskers. And I saved marine reptiles for the last. Due to time constraints, I will only talk about sea turtles, which are, contrary to fish and birds, an excellent substrate for diatoms. Of course, not all diatoms, but only those who can tolerate such an intimate relationship with a turtle. As diatoms on sea turtles have been my top research topic for many years now, I will do my best not to skew this presentation too much towards the sea turtles. Although, on the other hand, it is true that we now have quite a lot of data and information about sea turtles, so I could easily talk about diatoms on sea turtles for another hour. But let's try not to do that. Quite surprisingly, observations of diatoms on sea turtles, those very popular charismatic megafauna members, as zoologists like to call them, have only recently been made, as only 10 years ago, the first scientific articles describing sea turtle diatoms appeared. It did look like those papers somewhat reignited the interest in animal-associated diatoms. There were some new genera and species described from sea turtles, and maybe because it is relatively easy to sample diatoms from sea turtles, because sea turtles do come out of water on their own to nest, and while they are on land, they suddenly become rather slow. So maybe this fact, new sea turtle samples kept yielding new diatom species, and that sampling was easy contributed to how much we now know about diatoms on sea turtles. By now, sea turtles are well-known hosts for epizoic diatoms. Unlike fish, they do not produce mucus. Their skin and carapace are dry, and unlike birds, they have limited ability to clean their own skin or carapace. So turtles also do not possess glands to secrete oils or other antifolding chemicals. The skin and especially the carapace show relatively low enzymatic activity, and shedding, which is continuous, in most species is uneven, infrequent, and slow enough to not disrupt the already established diatom communities. Indeed, all 7 species of sea turtles at all developmental stages, except for, of course, hatchlings, host epizoic diatoms, often in great abundance. We are now pretty convinced that some diatom taxa, species and genera, are typical of or even exclusive to sea turtles. For example, genera like Chelonicola, Pulinia, and Melinella were first discovered on turtle shells and skin and appear to be part of a turtle-specific diatom flora. The mature biofilms on healthy sea turtles always contain at least 30% of the so-called truly epizoic sea turtle-specific diatoms. And since I said this, I now feel obliged to explain what those truly epizoic sea turtle-specific diatoms are. Well, they are those diatoms that in nature live only on sea turtles, occur on sea turtles frequently, regardless of the geographic location, and their abundances exceed about 25%. So in other words, species whose dominance is clear, as that would suggest that these species are adapted to the sea turtle substrate and that they not only survive on their animal host, but actually find their optimal growth conditions. For a list of sea turtle-specific diatoms, as we know them at the moment, Of course, the list may be amended in the future. You can check this paper that you can now see in the presentation. The article also proposes several testable hypotheses for why we actually are able to culture and maintain most of the seedorol-specific diatom strains in the lab, because we are able to do that. And that is why we were able to barcode those diatoms. Very different, distinct diatom communities live on both the seedorol skin and carapace. Is this a surprise? Well, when we think about it, it makes a lot of sense, because in the end, carapace and the skin are quite different from each other. They are different physiologically, biochemically, and also when it comes to their microtopography. The attachment points, for example, the availability of attachment points will be different on the skin and on the carapace. But seedorals, and in fact many other marine megafauna themselves, carry a lot of different living substrata on their bodies. And those living substrata, the macroamphibians, the invertebrates, are often host-specific too. Examples include seedoral-specific microalgae, sponges, and barnacles, or whale-specific crustaceans. And as we already know, those macroamphibians, the invertebrates, often create their own particular microhabitat and host their own specific diatoms. There are many more very, very interesting discussions that I could now start based on what I said here. For example, the dispersal, connectivity, and evolution of the especially host-specific diatoms. But I know we do not have time anymore, and maybe we can develop some of those follow-up topics on some other occasions if the audience is interested. I thank you very much for your time and attention.
Wow, fantastic. That was an amazing talk. Roksana, are you with us? I know, so this, for anyone that came in, this was a recorded session. Yes, you are, wonderful. Yeah, I just really appreciate the background story on how you came to this. I really had no, it was really helpful to understand stepping through colonizations on plants. You're on mute still. How does that happen? It doesn't look like you're on mute.
Is it my microphone?
Maybe, oh yeah, maybe you just need to turn the sound up a little bit.
Can you hear me?
Yes. Even a little loud. If it, maybe you turn it up a little more. It's like you're a little soft.
So I haven't done anything. Maybe it's
doing... Okay, there it is. That's great.
Okay. Maybe it's adjusting automatically in some way. I have this new camera set and it has a microphone built in, but it's been tricky. So I wanted to apologize for being late today and for canceling the other day. I've been having a particularly overcommitted year. So I'm really happy that I actually pre-recorded this talk. I knew it's going to be tight.
Well, you planned it ahead very well. Thank you so much. Let's see. If anyone has any questions, you can raise your hand or put it in the chat for Roksana. Tom, go ahead.
Hi, Roksana. Excellent presentation. I learned something here today, that 2-day turnover for the skin on the dolphins is amazing, and that certainly explains a lot of what we see. So I'm wondering, there seems to be a lot of opportunity to study succession. Possibly with some of the sea turtles showing succession, and some of these other substrates in animals that don't turn over as quickly, just being limited to these, possibly R selected diatoms. So just wanted to hear if you had any ideas about succession.
So you mean a succession in general on animals or on turtles?
Yes, yeah, on all of them.
So as you can see, maybe you also collected some of the invertebrates, right? So, or there are quite many studies about diatoms on zooplankton. They have very short life cycles, and you will often see that there's only one species on those copepods or other zooplankton members. So there's no succession in those communities, right? Because there's the life cycle of the copepod or the different stages of the copepod actually don't last enough for the diatom community to develop. There will be often monospecific communities of diatoms, but you can see succession on sea turtles, although at the same time, and I think this is why we know and we feel that these certain diatom species are specific to turtles, because at some point the community doesn't change anymore, right? On adult turtles, the community is stable. The part of the community changes because it's related to the location where the turtle is, but there's this, let's say, core community. Some people don't like the name core community, but let's say like this, that remains there, because those diatoms are just adapted to the turtle behavior, and they endure anything that happens during the natural life of the turtle.
Okay. Great.
Oops, you went over to mute, Tom.
I did go to mute. Thank you, Roxanne. It's great to see you again, and we'll talk more later. Yeah, great presentation. Glad you're here. Thank you.
Mark.
Yeah, that whoops, one more. There we go. Oh, almost got the video on. There we go. Yeah, neat presentation, Roxana. Just amazing to. I'm not a marine biologist, so it always just blows my mind to see what the diatoms in the marine world are doing that is so crazy compared to the freshwater world. I had two questions for you, if we can I ask two, Sarah?
Oh, yeah, go ahead.
Number one, let me just, you showed a picture of a whale that had a whole bunch of diatoms, I assume down by its bottom, by its butt. And is there any zonation that is associated with where those diatoms are growing? That, I mean, I would think, like, if you live by the butt, it might be a good place for nutrients. Does that happen with some of these marine mammals?
In fact, this is something we would also like to know, because we sometimes, Tom knows that as well, we sometimes get a chance to sample a whale, but it's a huge surface, and in darker whales, you actually don't see any colors. Even if there are the diatoms, you can't really see them.
Sure.
And you can't really brush the whole whale. But the truth is, when you go for the literature, it's very random. It's probably, again, a sort of a founder effect. When the inoculation happens, then you have, let's say, outbreak of diatoms, and then they slowly grow through that spot. And at some point, they may reach the whole body of the whale. But again, if the whale then moves into warmer waters, the sloughing becomes very fast, and a lot of diatoms are lost. So there's no zonation now.
Okay, interesting. The other question I have is just sort of, I keep, when I hear these studies of things like this, I know there's, opportunistic epibionts, and there's, does the turtle get any benefit from all this? Is it better camouflaged? Is it, I would assume it can't swim as fast with all that stuff growing on its back, but is it something like that? Like it's camouflage or something that is helping it, or? Do we know?
So turtles will have a lot of other epibionts, and the layer of diatoms will actually be very thin. So it's unlikely that diatoms alone would affect, the drag or the speed of a turtle or the weight of a turtle. But it may be that diatoms condition the biofilm so that other epibionts may nest into the biofilm, like for example, barnacles. But this is also just a speculation. However, those diatoms that live on the skin of turtle, this is often just 1-2 species, Medinella or some Turcicolas. They really seem to be adapted to the skin. And we also have, I couldn't really show my pictures because it's unpublished, and I think this video will be published, so I couldn't really show you the good stuff.
Oh,
But
no.
we have scanning electron microscope pictures of the skin and how the diatoms, dwell underneath the skin scales. And those diatoms they are never too many because the skin sheds. So like they are in perfect balance. So they may actually do something beneficial. So what that could be, they are pigmented, right? And sea turtles do get skin cancer. So perhaps they protect from UV radiation. At the same time, the diatom frusciule also was shown to change the wavelength. So maybe that benefit for the turtle, but also just diatoms are there, so they take the space, they produce their own metabolites, and they may protect the turtle from other pathogens simply because there is no space, because they don't want other microbes to take over, right? So it's like the natural microbiome of turtles. It pretty much looks like that. And then there's maybe even a better example from captive manatees in zoo. They only have one species of diatom, and when you look at them, they look very, very clean. So there is not enough diatoms, for us to see it. But when you take a sample, there's a whole layer of diatoms, and there's this just one species, which is Turcicola, which we think is manatee specific. So again, and it's aquarium, so there's actually a lot of other diatoms around. There's a lot of diatoms on the aquarium walls, right? So there are divers that would be actively cleaning the walls, but then there's nothing on manatee. So maybe those manatee-specific diatoms compete with those other diatoms, and that's why they provide some sort of benefit for the manatee, so that other microbes may be less beneficial or not neutral manatees cannot attach.
Oh, thank you very much.
Yeah, we have a question in the chat that I'm not sure if it's related or not. I'll try to say this word. Fibril papillomatosis occurs on sea turtles in polluted waters. Could it be due to the loss of diatoms? So
as far as I know, this is a virus, right, that causes skin lesions. So this is exactly what I had in mind. It may be related to the imbalance in the biofilm. So, for example, in the rehabilitation centers, the turtles are sometimes cleaned and the biofilm is completely removed. And then the skin is exposed to very random microbes, including some pathogens. So we could hypothesize that natural biofilm is a sort of protection from those other pathogens, but I don't have any evidence for that. However, it does sound plausible.
Okay, thanks. And Matt has a question. Go ahead, Matt.
Hey, good to see you again, Roksana. Nice talk. You mentioned something with the pinnipeds about whiskers and claws, and that sort of made me think about refugia. Particularly with the manatees and the whales. You get all this shedding and sloughing. How are the diatoms persisting and spreading every year with the the returns of the Arctic and whatnot? And I'm wondering about the mouth. The mouth is refugia, baleen, teeth, that sort of thing. I know we've looked at baleen and looked at teeth, and sometimes we find the diatoms and sometimes we don't. But yeah, I was just wondering if you had any other thoughts about refugia on animals for diatoms to persist past this sloughing.
Yeah, in fact, this is a very good idea. So I mentioned this very fast sloughing of skin in dolphins, and I did collect samples from
captive dolphins, and also zoologists confirmed that in warmer water and in zoos, the water is kept at a certain rather high temperature, they shed very quickly. So it's very unlikely to find diatoms on the skin. If there are diatoms, there are very few. But then teeth, they don't shed, right? And I think this first report from about diatoms on dolphins, it was, they were mentioning samples from the teeth, right? So yeah, I absolutely agree. Yes, there's a different chemistry, different physiology of a tooth, right? So yeah.